Welcome, ladies and gentlemen, to this VASCOPS webinar, How Dangerous is a AAA of 400 cubic centimeter in volume. First we're going to have a background section, then we're going to talk about volume as a risk predictor. After that we're going to see how deformable models can be used to measure the volume of aneurysms, before a biomechanical rupture risk assessment section will bring us to the first audience questions. After that, we're going to talk about the A4 Clinic software and see during a live case demonstration how this tool can be used to measure the volume of an aneurysm. That brings us to the second audience questions, which then will conclude our webinar. Now coming to the background, this is how the normal aorta is supposed to look like. However, in quite a few, specifically male cases, the infrarenal aorta develops an aneurysm, and as long as this aneurysm is small, the most important clinical question is how to monitor the rupture risk of these small aneurysms. The most popular method here is to monitor the diameter expansion with ultrasound. So plain ultrasound measurements are used to see how the maximum diameter expands over time. However, the study by Matuffi et al. In 2013 showed that this plain diameter measurement might be flawed. Their study was based on 90 patients having high-resolution CT scans from which three-dimensional models could be generated, which then were sliced perpendicular to the center line into 100 cross-sections, such that not only the largest diameter could be monitored, but 100 diameters could be monitored over time. This is one outcome of the study. This plot shows how the maximum diameter growth depends on the change of the maximum diameter per year, and since these two variables are not strongly correlated, we can conclude that the maximum diameter growth is not always seen at the site of the maximum diameter. Now, if you just see the change of the maximum diameter per year, let's say from plain ultrasound measurements, then you might stratify your patient into low risk, average risk, and high risk cases. Now, the study by Matuffi actually found that their fastest grower, showing 38% diameter growth, ended up in the low risk group. So that means that monitoring the maximum diameter cannot see maximum growth, and one really has to go for a three-dimensional point of view. Now, volume is such a three-dimensional measure of growth, and now we're going to discuss this measure as a risk predictor. To that end, we're going to look to the recent study by Lindqvist-Liljeqvist, presented at the IMAT meeting in 2014, where the authors based their data on 41 patients having a relatively narrow baseline diameter from 40 to 60 millimeter, because this is the clinically most interesting diameter range. Now, one key result of this study was that median diameter growth was 3.1 millimeter per year, so that's the expected measure and corresponds to 12%. Volume growth was 17 cubic centimeter per year, also correspond to 12%. So it's not surprising that the relative growth of these two measures are similar, because these two geometrical measures are linked. However, the more important results were that the baseline diameter could not predict diameter growth. So that meaning measuring the largest diameter of an aneurysm gonna not tell you how fast this aneurysm gonna grow in terms of diameter. Similarly, diameter growth, so how fast the diameter expands over time, did not correlate with increasing biomechanical risk indicators. And what biomechanical risk indicators we mean here, we're gonna see in one of the later sections. However, in contrast, baseline volume could predict volume growth. That meaning measuring the volume at baseline gonna tell you how fast this aneurysm in terms of volume gonna grow in time. And volume growth correlated with increasing biomechanical risk indicators. Some of this data has also been presented by Soto et al. At the ESVS meeting in Stockholm 2014, But now let's come back to the study by Liljeqvist and look to this ROC curve. So an ROC curve, receiver operator curve, gonna tell you about the information and content of a particular parameter. And in this first curve, we're gonna investigate the information and content of the diameter measurement. The question was posted here: can the diameter tell you if an aneurysm gonna end up as a fast grower, meaning being above the median growth of the cohort of the study here? What you see here is the ROC curve is almost diagonal, and that corresponds with the information and content of flipping a coin, meaning there is no whatsoever information in the diameter measurements. In contrast, looking to the volume, the ROC curve shows a very nice response here, meaning if you're gonna accept 25% false positive, you have a 100% chance to identify those aneurysms that gonna grow faster, meaning above the median of your cohort. Now this let us conclude that baseline volume, but not baseline diameter, correlates with risk. So that's the good news, but now let's come also to an accuracy assessment, how accurate actually volume can be measured. And to that end, we're gonna look to the cylinder of length L and diameter D, and we introduce an absolute diameter error, delta D. Now the relative diameter error can easily be computed by delta D divided by D, gives you the error in percent. Now this mismatch of diameter measurements also causes a volume error, and if you do the basic math here, you will see that the volume error, the relative volume error, is 4 times the relative diameter error. So that's an important conclusion, and we will come back to that later. But now let's go to the deformable model-based image reconstruction and see first on this movie how that works. So a deformable model, in this case is a 2-dimensional model, inflates within your image until it segments the body of interest, which is in this case the luminal area. Right. Now we're gonna use this segmentation technique to perform or derive a pre-segmentation of the luminal surface. That means slice by slice this 2-dimensional deformable model, also called snake model, is executed and gonna derive a pre-segmentation of the luminous surface. In a second step, we will refine this pre-segmentation by extending this concept to 3 dimensions, and then we call that the balloon model, and such a balloon model will be used to perform a segmentation of the luminal surface first, and then in a final step the segmentation of the exterior surface of our aneurysm. How that gonna work, we're gonna see in the later section. At the moment, it's just important that this technique also allows for manual interactions, simply because medical images are always full of artifacts, and it's very unlikely that the fully automatic algorithm will provide a high accurate image segmentation. Now coming back to the accuracy, the good news is that deformable models have sub-pixel accuracy. That means the diameter measurement will be better than the resolution of your image data. And that also explains why intra-observer variations are 1.5% for diameter measurements and only 3% for volume measurements in our approach. That really has to be compared to threshold-based segmentation used by packages like Mimix, Terraricon, and many others. And all these threshold-based algorithms require some kind of smoothing operations, and the typical smoothing operation is eroding away pixels on the surface. And if you do that with the typical object and image dimensions of our problem, you will see that you immediately end up with 10 to 20% volume error. So the conclusion here is that the segmentation is really the bottleneck of a volume analysis, and you have to apply very accurate segmentation methods in order to end up with reliable volume measurements. Now let's come to the biomechanical rupture risk assessment section, and this is the basic workflow. So we're gonna have our aneurysm, so our clinical problem here. We're gonna build a computational model. this model gonna predict something, and based on these predictions, that could be, for example, wall stress, we wanna interfere with our clinical intervention. Now the main difference to pure measurements is that these involves predictions. That means there are hypothetical numerical models involved that gonna predict something, and it's very important that these models are meaningful in order to end up with some meaningful result. Now, what are the biomechanical risk indicators? If you take a cylinder and put the blood pressure in there, so this blood pressure will be translated into tensile stress, and if we look to this tensile stress and take the highest tensile stress, peak wall stress, PWS, then we have identified the maximum tension in the wall, and that's already a first risk indicator, biomechanical risk indicator. However, rupture is not only dependent on the loading level, so meaning on the stress, but also on the strength of the aneurysm wall. And incorporating also this strength information, we can define a wall risk index, take the highest one, which is peak wall rupture risk index, so PWRI, and that relates then the tensile stress to the wall strength, and that is supposed to be a superior risk indicator. Now, how to analyze the derived results? Let's first introduce the average AAA patient by just plotting a diagram that shows you how this peak wall rupture risk index depends on the maximum aneurysm diameter. Now, this is such a diagram, and then you plot all your individual cases in this diagram and perform a regression. Then you will see that male and female regression lines look differently, and these regression lines reflect the average male and female patients. Having an average patient defined, we then can plot into our individual evaluation, into this evaluation chart, our individual patient. So this would be a case having a 50 millimeter aneurysm, which is well above the biomechanical risk seen in a typical 50 millimeter case. We can even go further and map it back to a rupture risk equivalent diameter, and in this case that would be 58 millimeter, telling you that the rupture risk that you see in this patient is typically seen in a 58 millimeter case. Now that would bring us to the first round of audience questions. Well, then now let's continue with the A4 Clinics software, and first we're going to look to the workflow. In order to perform these volume measurements or this biomechanical risk assessment, we need to have image data that allows for a three-dimensional reconstruction, and here is the image data that then runs into this semi-automatic image segmentation tool. Within this image segmentation, the luminal surface and the outer surface of our aneurysm are identified or segmented. This information is then used to perform a mesh generation, which is a required step for the biomechanical analysis, and in addition we can then immediately extract the volumes of our different types of tissues that are involved in our aneurysm. By prescribing the blood pressure and other patient-specific properties, we then can perform the structure and analysis based on the finite element method, which will then predict us stress in the wall, rupture risk in the wall, and also in the thrombus. Now we extract then the most important data from this analysis, such that we can identify the maximum diameter, peak wall stress, peak wall rupture risk index, volumes, and so forth. And finally we will have a reporting task where also other patient-specific information can be put into the final report. And the final report is then output as a PDF file, and how that looks like we're gonna see later in the live case demonstration. Now that's the basic workflow to perform this type of analysis, and now let's see how the system is integrated into the clinics. All the calculations are take place on a local terminal. So there you're going to derive your patient data, risk indices, volume measurements, and so forth. And if you just use the plain software, you can use Windows file system to store your data. However, you can also integrate the software into the VASCOPS system such that you can directly read DICOM data, so your image data from the VASCOPS system here, perform then your analysis, and then you can pack your analysis report, so the PDF report can be stored back in the VASCOPS system. However, not all the data you have generated, simply because the VASCOPS system doesn't support the data format used for this analysis. In order to account for this drawback, we also provide you with a database that runs locally on your terminal, such that you have all the features you are used to from a VASCOPS system directly locally at your system. Now coming to the data analysis and reporting steps. So here geometrical and mechanical data is reported in and output then in an analysis report. And here again, the individual case is related to the average patient using this evaluation chart here, where then the individual patient can be seen with respect to the average patient. Now that comes now to this VASCOPS diagnosis service. So if you are not willing to perform all these steps by your own, then you also can send us your DICOM data through a password protected DICOM upload platform, and within 24 hours you will receive your analysis report with all this information. Now let's come to the summary of this first part. We have seen that volume measurements in AAA surveillance have several advantages over diameter measurements. So that's the clinical news. However, you have to be careful because volume error is 4 times the diameter error. That means you really need very accurate 3-dimensional reconstruction schemes. And A4 Clinics uses deformable models that known to have a sub-pixel accuracy. And then these 3-dimensional reconstructed models synergetically combine with the biomechanical risk assessment, meaning 5 more minutes spent in time will also provide you with a detailed biomechanical risk analysis. Well, brings us then to the live demonstration, and we're gonna see how we can measure the volume of a particular case using the A4 Clinics system.